Hello, everyone. You guys can hear me well. I hope so. Otherwise, please let me know in the comments. We've tested it, so it should be working fine. And be very welcome to our first English talk that we've been doing through the Wild Place, and and special welcome to some of our friends from other continents. We have different people here from India and from Nigeria that were very interested in this talk. So we're doing this in English to try to reach this people in other parts of the world. So let's see let's see if they like it. You guys please give me the feedback. And well, let's see also what we're gonna talk today. Today's talk is going to be about amphibian medicine. So, the purpose of this lecture was actually to try to give uh, people abroad a review of what we know about the amphibian medicine currently, and also to try to spread the word on why veterinarians are being so important within the amphibian conservation in itself. So I hope you guys understand my English, and if you have further questions about any sort of the language or the words I've been using on this lecture, please send us a message and let us know. We will be like very happy um, to answer all of your questions, okay? So, before we start, amphibian medicine has been like a topic that has not been very discussed. And due to that, we usually kind of like renegade amphibian medicine for most of part of it. And although in Brazil, we're seeing an increase of amphibian pets right now, we out abroad, especially in Europe, in the United States, amphibians have been had as pets for a long time, and most of the knowledge we know we bring from them. And what we try here is to use that knowledge and further develop it based on our experience, especially because we have so many different species here in South America, especially as you guys from India or from Africa will have different species as well. So here we will try to give you a full introduction on what the amphibian medicine is and what it could be, especially having more people involved. So, if you don't know me, my name is Iago Junqueira. I am a veterinarian and I am a master in animal science, and I am working as a professor at the Wild Place. The Wild Place is a business that has courses, and we are trying to do education for conservation, not only teaching all kinds of people about the importance of conservation in itself, but also trying to professionalize people to work with conservation medicine, conservation education, and anything that can help for conservational purposes, not only the animals in itself, but also society and the world we live in, right? So we have a big mission and a lot of passion. We are made with a team of a lot of students from majors as biology and veterinary medicine here in Brazil, and we very welcome any student abroad that would like to be part of it. Um, I'm also the only veterinarian at the Brazilian branch at the amphibian specialist group. So if you don't know amphibian specialist group, it is a branch of the IUCN, the International Union for Conservation of Nature. Whenever you see that a species is endangered somehow, and there's like different categories for it to be endangered, such as like critically endangered or vulnerable or something like that, who sits down to try to put that up is a group of specialists. And these groups of specialists, they have like a main group, which is for us, for example, would be the amphibian specialist group globally, and then there's different branches of it to specialize in different species in different areas of the globe. So I know that they have like a very strong branch in the United States, in Central America also, and in Europe and in China. Not sure about the Southeast Asia, South Asia, and in Africa, but I know they are starting and they're getting together to put their own specialist groups too. And what is an unfortunate is that most of these groups won't have veterinarians, and I blame us for that because we as veterinarians we usually don't focus on a greater view, on a greater perspective of how can our knowledge help conservation purpose in itself. So what I want to bring here is a little bit of this view that how can we be inserted into it, how we can make a difference, and what what is the need for us within conservational fields, and how amphibian medicine in itself as a branch of the knowledge within amphibian conservation can be quite a useful tool to save different species. So, if you don't follow us on social media, please do. We have the ASGBR page on Instagram. We also have the Wild Place BR, which is for Brazil. Also here, tagged on the top. And there's my own personal page too, Vet Iago Junqueira. You guys can follow me there as well. And we always giving different kinds of opportunities of learning, not only within amphibian conservation in itself, but we're also trying to fill out different gaps that are usually within, especially within the veterinarian formation as a professional, because when we are in vet school, there's a limited amount of time that we can spend like learning different disciplines, and wildlife medicine in general, it's not like being a full subject that's giving like a lot of time within these formations. So we try to fill those gaps and be the tool that will help you to get prepared to work with wildlife wherever you are. So please follow us and let's get the lecture started. Uh, oh, I have a couple more um, advices for you guys, and one of those is that this class is being recorded, so you guys can re-watch it on our YouTube channel in the future if you want to. You can also share it with your friends and with your colleagues, and for those who missed this talk. Another thing is that in the end of the talk, there will be a form that will be on our video description on there that you guys can fill it up to ask for a certificate that you've watched this lecture. The certificate has a cost, which I believe is $10, and you can like send it to us. This money will be used for other further educational and conservational purposes, and we will email you guys the certification just to, I mean, for credits or for whatever you might need this. So pay attention whenever I say the keyword, because the keyword will be needed to fill up that form. So that way I know that you not only is filling up the form because you want the certificate and you haven't watched the lecture, but that you did in fact watch it at all and participated in it. Okay. All right. So following up, where do we start? I believe that first way for us to start is telling you guys who the amphibians are. These amphibians, they are all vertebrates within the phylum Chordata and the kingdom Animalia, and they are classified within a class called a superclass first, called Amphibia, and within the class superclass Amphibia, there is a class called Lysamphibia. Lysamphibia literally stands to amphibians, and this prefix lys means smooth-skinned. So they are amphibians with a smooth skin, and these class comprehends all the currently living amphibians that we have in the world. These amphibians, They are further separated into three orders. The first order is order Anura, which literally means without a tail. There's another one called Coldata that means with a tail. And the last one is called Gymnophiona. And Gymnophiona means naked skin like animal. So we have three kinds of different amphibians that are divided into these three orders, and they have their own characteristics to themselves. But some characteristics are like for all of them. A general character that will like describe all the amphibians. First one is that they are poikilothermals. That means that the amphibians cannot regulate their own temperature, which means that they need the temperature of the environment to control their own. And that's a very important information because whenever a veterinarian is used to work with, for example, mammals, and they are getting like an amphibian in the in for like a checkout or something, we don't even need to take their temperatures because their temperatures will be the environmental temperature. So that also gives us clues. For example, if we want to give them medicines and we want them to metabolize those medicines very well, we have to heat up not only the animal in itself, but to heat up the environment because that way their bodies can adjust the temperatures to what the environmental temperature is and can get their metabolisms like with a boost and get them faster and metabolize those meds faster too. If we do the opposite, they will take a long time, for example, to recover from anesthesia. So those are things that we have to be aware and have as a veterinarian, and we have to put out there or as the knowledge to avoid that other professionals or even like if non-vet professionals that may take care of these animals that they are so sensible to temperatures and also to humidity that changes or especially very like hard and fast changes into this kind of environmental characters can cause a lot of problems for us having these animals as a patient. If you pay attention to the map that I put out there, the red ones show us where there is the biggest richness of amphibians around the world. And wherever it's gray, it means that it does not have amphibians living in there. So we can see that the largest amount of amphibians are around the Amazon forests and the Atlantic forests on the coast of Brazil. We cannot see it very well, but there's like a orangish, yellowish patch also around Africa, especially within like very close of the to the equator line. This equator line is where it we have like the wettest and also the warmest places on Earth, and that makes sense because that's where amphibians thrive. Whenever it's whenever they have like a very cold environment such as the Arctic Circle or Antarctica, we won't see amphibians in there because it's too cold for them and their metabolism cannot like adapt to just such a harsh environment. But the same thing happens with deserts. Amphibians need at least a minimum amount of moisture, and although some amphibians may live into arid regions, whenever the this humidity is too low and they can get like low and cold, especially during the night, they might not habit in there. So we have patches of the Sahara Desert, but also the Arab Desert and the Mongolia Desert that won't have many species of amphibians if they had amphibians at all. In South America, that happens also to the Atacama Desert and the Tierra del Fuego, which is the further southest part of Brazil, of not Brazil, but South America. And if you see Australia, they also have like a south patch that they don't get amphibians in there because of its aridness and low humidity that those places can get. So amphibians and temperature are fully connected, and within this connection, humidity plays a big role in it too. For amphibians in general, we have over 57, like, we have exactly 57 families, 468 genera so far, and up to date we have 8,942 species. And those species, they change every day because new species are being discovered, and unfortunately some species are being extinct as we talk. So that kind of like rings a bell for us, like to try to understand why are these animals disappearing and being extinct. And some of them might even involve us, veterinarians, and our lack of action. So that's a call out for us because as amphibians are nowadays the most endangered class of vertebrates, with like over 41% of the species having some sort of classification within endangered, even though it's like vulnerable or endangered itself or critically endangered or even extinct in the wild. That gives us like a very bad look on the situation that amphibians are getting through the world. We also have like a big amount of amphibian species. They are classified as data deficient. These animals are even more problematic sometimes because if it's data deficient, it means that we have no clue on what's going on with them. Some of them are data deficient because they're so hard to find that they haven't been seen for many years. So we cannot like put up if it's already extinct or if it's just like the lack of data. Some other data deficient species, they are also animals that sometimes are being they they live in such hard areas to get into and to do research that we cannot like get collection data from them. Some of these places are, for example, the Darien Strait in between Colombia and Panama, where they have so many severe conflicts within different groups of people that it's critical to do research in there because there's so much to be discovered, but it's not being allowed because these groups of people, especially terrorists in itself, they do not allow anyone to go into the jungle, and the jungle itself is dangerous as well, so it makes it like very hard to understand the situation of amphibians in there. Same thing applies to other parts of the world where people conflicts might cause impact on it. Even the country's politics can have like an impact on it too, because whenever we have a country that might have trouble getting their own people to study a group of animals, sometimes people from abroad wants to go there and try to discover their that those sort of informations in order just to make science, but also to try to help those animals. But the politics can get things a lot harder. So amphibians in itself have a lot of species that are threatened, and that's the message I want to put out there. Other characteristics of amphibians are also that they are scaleless. That means that they do not have those hardened patches that cover their bodies, just like the reptiles have or that birds have in their legs. They also are thin skinned, which means that their skin, compared to what we know about mammals, for example, it's a lot thinner, and all the different uh, all the different patches of skin, like the different subdivisions of the skin, are very thin. The derm in itself is quite thin, and compared to mammalia, for example, compared to mammals, it has a very large amount of vessels. There are capillaries that disperse within the derm. They also have a very thin epidermis, and as us human beings, we have usually around like 30 layers of keratin. Amphibians only have like literally one layer of keratin. This is important for amphibians because this thin skin helps them two ways. First way is that the the thin skin allows them to do uh, skin respiration, and the gases that spread out through the capillaries within the dermis, they are so close within the atmospheric gas that they can literally get pulled out by attraction and exchange gases just to get like very uh, the amphibian blood very rich into oxygen, and they liberate this CO2 into the atmosphere. Another thing is that whenever they're submerged in water, they can also change different elements that they might have into their bloodstream within the environment in itself, the water environment in itself. And that happens due to difference in the concentration of the solutes they are in the blood and the solutes they are in the water. So, for example, if an amphibian produces within its metabolism a lot of ammonia, which is a result of any metabolism or cellular metabolism in an animal, the amphibians might have like a choice of, if they're in the water, they can eliminate this ammonia without having like any energetic cost by changing it, exchanging it with the environment and the water itself, if the water doesn't have a large concentration of ammonia. So because of this different concentration, it loses the ammonia to the water where it's immersed without any energetic cost. And that's very good for the amphibian. If it's in the, I'm sorry, I have dogs in here, so if you hear barks, it's just them. Um, if we have an amphibian in a terrestrial environment, it might need to convert this ammonia into urea and then excrete it through the kidneys. But the water helps the amphibians with a lot of aspects, and some of them being those within the exchange of solutes. Even oxygen can be exchanged within the water. If the amphibian is submerged and the water is oxygen rich, it doesn't need to go all the way up to breathe. It can exchange the oxygen within the water he's immersed into. So those are very good characteristics for amphibian survival, but those are very also important characteristics for the veterinarian to know, because we can use these characteristics in our favor. Parasites and diseases also use those characteristics in their favor, though, and they can cause trouble to the amphibian, just messing up with his skin. Amphibians also are known to usually be semi-aquatic animals, and this connection to the water is not only linked into their ability with the water, like with the exchange they can do with the water and how the water environment helps them a lot, but the connection between amphibians and water is also needed to reproduction. Amphibians are usually near bodies of water because their reproduction is usually external. That means that most amphibians would put up uh, their spermatozoids and their ova into the water, and fertilization will happen outside the body of the animal. And the water where they are immersed into, it serves as the channel to link this kind of connection between the spermatozoid and the ova, as a way of getting from one point to the other. So whenever we have amphibians that live very away from water fountains, they usually have different strategies to try to cover that up, but they need at least a little bit of moisture. Their eggs also are very sensible to water loss, and as they don't have like a shell around them, that makes them very vulnerable not only to predation but also to infections such as fungal infections of the ova that can kill all the fetuses, but also within the dehydration of the ova can cause a lot of trouble too. It can kill the fetuses within the eggs. So whenever amphibian is ready to hatch, it as it doesn't have like an egg yolk within the egg, because that only started to happen within the reptilians, amphibians, they usually have like a larval form. This larval form is the tadpoles that most of us know, and it usually have completely different, not only anatomy, but completely different physiology than the adult form. So metamorphosis, I usually like to my students to compare like it's not like we have like a rat that's very when it's born, it's like a pinky, hairless little chewing gum that grows into having hair and all of that. But imagine if that little rat also have like a different way to breathe, a different, completely different anatomy of their digestive system, a completely different anatomy of their excretory system. So it the tadpoles, they have to pass through this metamorphosis that looks like they're just like growing up, but they are passing through so many substantial changes within their whole metabolism, their whole different tissues, that is classified as metamorphosis and not just development in itself, like a simple development in itself. And they have to gather their own resources too. So mother frog or mother salamander, most of the time they won't provide their offsprings with anything, but oh, some Gymnophiona do. So we already know that Sicilians, some species might shed their skin often whenever they have like this little Sicilian to feed them with the old skin. So we have some parental care. Even in frogs within the Anurans, we have some parental care too. Some frogs can carry their tadpoles around and change them from pound to another pound whenever the water is like in low quality or it's drying up. Some of them can carry their eggs. We have males that carry their eggs into their vocal sacs, which is like an aperture. They have like an aperture uh, to a vocal sac on the male that's right besides the tongue, and then they can put the eggs in there for them to be protected as they hatch. Some species can put their eggs within the back of the female, and then they, as the skin grows up, it covers up the eggs, and the eggs are literally attached to their back. And whenever they have the hatching process happening, it's like the babies are coming out the mom's back. So we have different kinds of strategies for them, but the water will be always enrolled in it somehow. Okay. Well, and why are amphibians threatened though? There are some species of amphibians that are threatened by emerging diseases. Emerging diseases are currently mostly two diseases that we're talking about. One of them is the ranavirus, which is a virus that also targets some reptiles and can cause disease in reptiles and even fish. But amphibians are very susceptible to it because the this ranavirus, what happens is that the virus looks for endothelial cells, and it usually multiplies inside these endothelial cells and kills the cell. And when it kills the endothelial cells, it can open up like a little hole into the vessels, the walls of the vessels. So the blood that's coming up within the vessel, whenever it finds a hole, the water of the blood can go through that hole and cause edema in the tissue that are around it. And if the hole is big enough, not only edema can be caused, but the actual blood cells can go out of it and they cause hemorrhages. So it's very common for amphibians that have not only to have within rhinovirus, not only cases of edema, even if it's local, regional, or if it's generalized like in Anasarca, but it can also cause hemorrhages in different scales too. Another one is the BD, which we call BD, and that stands for their scientific name, which is Batrachochytrion dendrobatidis. This is a fungal infection that they have like a different family of fungus called the chytrids. In this chytrid fungus, they are everywhere, but and they don't usually cause trouble to anybody. But for amphibians in itself, they started being some problem because this fungus likes keratin, and everybody has keratin on their skin. But as we have a lot of layers of them, whenever a fungus comes like into our skin, for example, like a chytrid fungus, it doesn't cause much trouble. It just makes its little cysts and goes away. But within frogs, as they have only one layer of keratin, they started getting like some trouble because this layer of keratin, although it's like a simple layer, it's very important to them. So whenever the fungus finds a frog, and it literally finds a frog because it can swim, this fungus has like a free stage form that's called the zoosporidium. The zoosporidium has like it looks like an spermatozoid somehow, and this little sperm like fungus, it's on the water, on the environment, wherever there is some sort of humidity. If it has a little bit of water, it will live in there. And whenever it finds the frog's skin, it goes into their skin and it makes a cyst called the zoosporangium. The zoosporangium, it grows until it cannot grow any further, and then it blasts. And whenever it blasts, it liberates other zoosporidium, which are the mobile form of it, into the environment. These new free forms can infect like the cell like right next door in the same amphibian, or it can spread to other amphibians through the water environment. So what amphibians get from that is that whenever there is a new zoosporangium being formed, there is an inflammatory reaction to that. And the immediate response from the amphibian is to put up a new layer of skin, like we shed that skin and that fungus will go away, right? Not right. What happens is that the fungus, it goes further into the new layer of skin, and it literally anchors the old skin within the new skin, and it prevents the amphibian to shedding. So the amphibians try to shed again and again and again, and then these multiple layers of old skin start to get stuck into the amphibian, and not only they start like to degrade and get infected, like just for being like old skin that's not irrigated and that the amphibian don't need anymore. But it also makes the layer of skin, especially the epidermis, way thicker, and it prevents the amphibians from breathing through their skin or to change those osmotic relationships that I told you guys about within their environment. So they would either have to spend a lot more energy to lose a lot of the solutes that they have in their blood to excrete the excess of it. And sometimes it's like not even possible. So they intoxicate with their own solutes, or they can like have such an stressful event within not only an osmotic stress, but also a hypoxemic stress because they're not exchanging oxygen very well, that they start having like a literal metabolic collapse. And with that, their immune system literally goes like so bad that it cannot prevent simple infections that he usually can defend themselves from happening. So the own bacteria that grows in the skin of a healthy amphibian usually starts to spread into an amount that the amphibians cannot handle and control their multiplication as well, and they start getting septic. So these amphibians start having like a skin problem that blocks them for doing very important exchanges with the environment from like water to oxygen, and that stresses them. It reduces their immune response, and then different secondary bacteria or either fungal infections can happen, and those can become septic, and that's what kills the amphibian. And that can happen really fast, like within 10 days, an amphibian can get exposed to it, develop it, and get so critical that it dies. So this diseases are very important, and we're going to discuss a little bit more about them in a little bit ahead. There's also species that are being threatened by habitat loss and habitat quality loss, which are two different things. Habitat loss is that there's like a jungle and we cut that jungle off. There's a forest, we cut that forest off. There's a swamp and then we literally destroy the swamp and cover it with sand. So that's habitat loss. We literally don't have the habitat in there anymore. And whenever we have habitat quality loss, that can be as bad as the habitat loss itself because it gives us a perspective, like a perspective that the habitat is still there. But sometimes, even like if a forest is there, it doesn't mean it has a good quality within their ecosystem that it's enough to sustain the life of amphibians. And I mean that by an example. If they have like a protected area, which the water stream comes from outside of this area but passes through this area during its current, if I have some sort of pollution, like before it enters into that protected area, it can contaminate the water within the protected area. But from outside, we'll look at it and say, well, the forest is doing well, like the environment is protected and we're doing good. But what happens in there is that this pollution can cause the water quality to this whole environment to get so bad that even if the plants can survive, most of the animals they are dependable of the water quality in itself can die, and that will cause further imbalance into the ecosystem because species dying will lead to other species not having what to prey off and going away or dying too, or starting to prey on different animals they are not used to prey on. And some of those animals that used to be preyed by those animals that are dying, they start to accumulate and overpopulate those areas. So it's common for us to see that here in Brazil because sometimes we have like a river that crosses through a city and then it goes into a forest. And when we go into those forests, the population or diversity of amphibians in there is so bad, and we don't understand why because the forest is being protected in those areas. But that has to do with what happened before the water crossed into that border, because now the water fountain of this whole environment is in poor quality. This quality may be enough for the plants, might not be enough for the frogs. And then with these frogs die-outs, we usually have spread of arborivosis, for example, because there's a lot of mosquitoes that will multiply, benefiting of not having their biggest predator, which will not be the frog in itself, but their larvae. The tadpoles are very efficient predators for mosquito larvae. There's also ecosystems that have been fragilized because of the intense loss of biodiversity and also because of invasive species. So this environment, sometimes they even have invasive species that can worsen the problem. With areas they are close to urban sites, so peripheral cities that have like islands or islets of forest around them. Sometimes the invasive species within these patches of forest, they are so high in amount that it prevents the ecosystem from happening because the animals are being overpreyed. The animals that used to live in that area, they're being overpreyed, or even they are competing with the invasive species somehow. Some of the invasive species that caused a lot of trouble are usually the cats. Cats are very good predators and they will prey on birds, they will prey on lizards, they will prey on snakes, and they will prey on frogs. If it moves, they will prey on and they can cause a lot of trouble with frogs, especially with small populations of frog. If a cats get inserted into an area where there's like a frog that's very endangered, it can literally, like in a month or two, just one cat can decimate the whole population of frogs, or even it can kill specific specimens that were important for conservation purposes, such as reproductive females that get more lethargic and they're easier to prey on. So those kinds of invasive species are a very bad problem for amphibian conservation too. But there's other amphibians that can be a problem for amphibian conservation. An example of them is the bullfrog. Bullfrogs not only eat other amphibians, but they also can be like a spreader of disease within the environments they go on, because they can be very resistant to some diseases that affect amphibians in general. By like, they don't usually die, but they carry the diseases around and spread them into populations where they're not supposed to be around, and those populations usually don't have the immune capacity to fight those diseases, just like the bullfrog has. Um, Sometimes we have also ecosystems being fragilized by unbalanced anthropic activity. What happens is that, especially in places like Brazil, we have large plantations, especially in monocultures, we call them, where for like a large amount of space they plant only one plant. There's like a one species all around, like a lot of hectares of plantation, and the these plants having like only one single species of plant in there, they cannot create environments where multiple species can prosper. Amphibians are an example of it. So as they cannot have the environment within the plantation, they won't try to cross it and they won't inhabit it. So even though sometimes they can try to find their prey in there, they won't go into it because they know that if they go into it, they might get literally lost within the field or the maze of those plantations. So they keep themselves into fragments of forests. They're sometimes maintained by law. Here in Brazil, for example, if you have like a large plantation, then there's like a certain percentage of land that you have to keep intact and as a protected area. That doesn't solve the problem in itself, too. It just creates literally islands of where amphibians can live, and those whole harvestings that we see just works like a huge sea that the amphibian cannot cross, that isolates the populations into little inlets of and group different like small groups of frogs, and that can lead to inbreeding, that can lead to habitat loss, and they can and uh, that can lead to habitat quality loss too, and that can even makes the amphibians susceptible to different changes of the environment that might happen in a smaller area, because smaller areas are very susceptible to changes on temperature or humidity, or because of their small amount of area, they can, if like a wildfire catches, it can destroy the whole area, or if this area is close to urban sites, it can get like invasive domestic species too, and there's a lot of consequences to that. All of this might reflects into economic and health reflexes. And how so? So without the amphibians into an environment, or without like most of the species in an environment, if a species goes into extinction, for example, into a forest, it usually it filled a place within we call like the ecosystem network. So one species has multiple connections to different species within the ecosystem. It's like a neural system where everything is connected somehow with another species in some level. Some of them, some of these relationships between these different species, they are ascendant, where these animals in the top of the net, they eat these animals they are in the middle or lower places of the net. And these animals in the middle, they might prey on the animals that are on the basis of that net too. And all these animals are interconnected somehow, right? I said that. So if I remove one of these connection points, which would be an species, I leave as a blank a lot of areas of connections that were within this ecosystem before. So leaving those gaps in this network makes the network got unbalanced, and that can make the basis of the network to increase in volume and the population, and the top of the of that network will shrink because they don't have enough to prey on. And the amphibians are right in the middle of this network. So whenever one species is fully, completely removed from it, these gaps that the amphibians leave in those networks of environments, of ecosystem networks, they are a huge problem for the ecosystem as a whole. And as I said to you guys, some of those problems will be connected, especially with spreads of diseases into human populations, because they are very effective controllers of different vectors of different diseases for human populations too. One thing that's caught my attention is that one other problem we are usually seeing is the traffic and amphibians as pets and biopiracy. Trafficking is being very present since, like, we know about animal trafficking. They've been trafficking amphibians to many different places. Most of them go out of Brazil and go to Europe, United States, and China. When they go to United States and Europe, they are usually bought by collectors that want to have different species to fill up their collections, which I find like quite selfish, but they usually do it. They buy animals from the wild, from people that can literally capture them and ship them to these countries just to be part of a collection. And in China, we have a problem too, which is some species are known to the traditional Chinese medicine to have properties that they believe may cure some sort of diseases that they have interest in treating. So they are being like trafficked to those purposes too. Another thing is that within the internet, we we see every time more and more pages dedicated to frogs. They are getting like charismatic, and people are starting to like them very much, which I find that it's lovely, but it comes with some bad consequences too. And those consequences are due to um, the, how do I say it, due to inadequate management from people towards their frog pets, to say it in a light way. So what happens is that you look into your TikTok account and you find a frog video, and you see it that people put them into doll houses, they dress them up, they put accessories into them, they make stories about them, and they pet them, and they handle them with their bare hands. And all those things I just told you guys are things that can cause really real trouble to amphibians and their health. One thing that we do not notice is that how many different environments can impact the amphibian's health within the changes of temperature, within the changes of humidity, within the changes of substrate they're put on, the level of shade, the level of light. Everything influences the health of the amphibian. Whenever an amphibian is overheld or over manipulated, it causes a chronic stress that will lower their capacity to respond to as with their immune system. And another thing is also that anything that we put into the amphibian skin can cause changes into that skin that can lesion the skin in itself, or it can dysregulate the microbiome that the amphibian skin has and stimulates that bad bacteria grows wherever the good bacteria died, causing skin disease. And as we've seen, the skin is a critical part from the amphibian, so we try to avoid that in all costs. One thing that literally causes really bad things into the amphibian skin is handling them without proper protection. So handling amphibians can be very easy. Most of amphibians can't be held and can be managed, manipulated, or whatever you call it. But we have to think on two things. The first thing is that how this amphibian can harm me. Some amphibians have very bad venins that and poisons that it secretes into their skin and can intoxicate the handler, especially amphibians from the Amazon forest that we call the dendrobatids. Those are called also dart frogs because indigenous people used to usually pass their darts into the frog's skin whenever they're hunting, especially when they're fighting other tribes too, just to try to literally poison the enemy whenever the arrow or the dart hit it. So we see this potential being used by human beings for a long time, and it literally is a very well known uh, thing within frogs. All frogs have poisons. What differs from one frog to the other is that what's the effect that that poison will have on the animal where it's been inoculated or had contact to. Second thing is we have to think is how the frog can get harmed by us too. And not only the inadequate handling may cause trauma to the frog, but handling it without the gloves. We we are not aware of how many chemical compounds are in your hands. Whenever you go to the bathroom, you go and you use the bathroom, there will be like different droplets of things, different secretions of our body that might still be in your hands that can be toxic to the animal. But even if you're a very hygienic person and then you go and wash your hands with soap, the soap can also be very bad to the frog skin. And although you don't see it, microparticles of it stays on your hand forever. Like you just cannot get rid of it, especially if you keep doing the washing your hands like for a long time or multiple times. And with us veterinarians, when we are in the clinic, we try to hygienize a lot of times our hands when we use different compounds from soaps such as chlorhexidine, for example, or up to different desanitizers as alcoholic uh, compounds and alcohol gel or whatever, all those compounds are also toxic to the frog skin. And even if you just like took a shower in the morning and you wash your head with your shampoo and you put some perfume, the perfume is usually alcoholic and it stays on you for a long time because of those alcohol compounds. And the shampoo you've passed on, it has the chemicals that can harm amphibian skin too. Your solar screen can have chemicals that can be bad for frog skin too. And you know, you get a solar screen to put on, you use your hands. And although you're passing it on your body, a lot of it stays in the palm of your hands. And if you look at the at the product, it will say like that it stays for 24 hours, it stays for 48 hours, or even longer. So Whenever we do it barehanded, any kind of manipulation of amphibians, we're putting all those chemical compounds into their skin. And that can lead to what we call uh, skin dysbiosis. And the skin dysbiosis will lead into dermatitis. Not only the chemicals in itself can cause trouble, but imagine if you're a person that do not use any sort of cosmetics. You still sweat. Your hand has so many different pores of sweat glands that you cannot think of, and you're not aware of how much your hands sweat. You have to keep your hands sweat because it's good for you as a person and as a physiologic body that you live in. But for the amphibians, those sweat that you have, the sweat compounds, they they have a lot of salt in it, and they cannot only dehydrate the amphibians you're putting your hands on, but it can also cause the dysbiosis. The chemical compounds that you produce, your sweat, is very osmotic for the bacteria that are in the amphibian skin. And they can literally make the bacteria, especially the good bacteria, to be destroyed and open up the space for bad bacteria. They're usually very resistant to colonize those spots and cause dermatitis. And you might not see it at the same day. So dysbiosis may occur like for the next 3 days. So what we usually see is that a lot of biologists, for example, that goes into the field or into the jungle and they collect amphibians, they hate using gloves. So they don't use it because they say that they've never seen an amphibian get like anything from not wearing gloves. But that just happened because it doesn't show the signs in the same day. And biologists usually take the frog, take their measurements, their samples, and they release it. And in the next 3 days, that frog can get like a very bad dermatitis, and it can evenly die from sepsis, depending on the immune characters that might have at that time. So it's not only because you're not seeing it right away that it's not happening, and we have to be aware of it because as veterinarians, there is a ethical code that we follow, and within that ethical code there is a couple of things that we have to pay attention because we've made a swear on it. One of the things is that we cannot be negligent. And whenever we do not use gloves, now knowing that that can harm the animal we're caring for, that's negligence. Therefore, not wearing gloves is unethical. So another thing is that we cannot be imprudent. Not wearing gloves is being imprudent, not only to the frog, but to yourself, because you can also get poisoned depending on the species you're working for. And then there is imprudence, and the other one is Imperitia. I don't know how to say that in English, but when it happens when you're not a period on something. So if you are working with frogs and you've never had any sort of training before, that makes you not a period on that matter. So you might act and cause more harm to the frog than goods. And that's unethical too. So we have to pay attention to those three things combined because that can make you uh, answer for a process on unethical procedures. So whenever we have a frog, we are now trying to bring this one health approach just to justify how we do need to learn about frogs and how frogs' health is very important within the One Health too. And when I say frogs, I can also try to open it up to all amphibians, even though the frogs are the ones that I work the most. So we have to think about human health, we have to think about the ecosystem health and the animal health, in which the veterinarians are a key professional. They are involved in the three different circles that connects One Health to make us deal with health in general. And one thing that it might not be like quite obvious for you guys too is that whenever we're talking about health, the opposite of health is not disease. The opposite of health is unbalance. Health means literally homeostasis, and homeostasis is the equilibrium, it's balance. So, whenever you are sick because there's a lack of balance. Your body is unbalanced somehow. It can be unbalanced because of the things you eat. It can be unbalanced because of the way you live. It can be unbalanced also because of a pathogen or pathogen. I don't know how to pronounce, but that's the thing. So, whenever there is no health, it's because of the lack of balance. And when we think about uh, a health of a being, even though it's a human or it's an animal, it's easy for us to understand when something is unbalanced, that the animal or the being in itself it's sick. We can look at a plant and see that it might have their organism with imbalances and might be dying, and we can say that it's sick too, that it lacks health. But we most of the time do not see the health of the collective. And how should we do that? So I'll try to open up your guys' minds for like trying to see society, like the city you live in with all those people and animal pets and all of that, that's society as a one living thing. So what I'm trying to say is that sometimes when there is a collective of organisms, it's like they have a collective health, and different kinds of things can cause this health for the whole society to be unbalanced or to get like a disease. Diseases in itself are a problem, but if you have like a unfair distribution of money or of wealth, you'll have people that are in poverty and people that are very rich, and the system is not balanced, so it will cause problems in itself. So this unbalance will make the whole society sick somehow. A society that's not balanced, it it will have needs for a certain amount of people that will prevent them on trying to keep the balance on, or even thinking of the balance of the other cycles. If you have like a group of humans, they are starving, that they don't have something to eat, they won't care about the ecosystem that we're trying to save or the species of frog that we have to save. In that matter, if they could, they would go and eat those frogs, and they don't care if it will be extinct or not. So that's also part of One Health. There's different problems within our society, although it looks like a lot philosophical. It's a matter of One Health too, because we have to. One Health is trying to get the balance of different groups of organisms back into balance. And societies, it can be seen as an organism the same way that the ecosystem can be seen as an organism too. So you remember that network I was talking about within the ecosystem, where plants and animals and all the living beings in there, but also the resources and even not live resources such as the minerals and water quality and the compounds and all of that is evolved to maintain the balance of the one ecosystem. And if one of those things are taken out, those ecosystems can go into unbalance. Therefore, they would be sick. If we have a species of frog that's being affected within that environment, and we remove that species of frogs that have a lot of connections within that network, that ecosystem that's now frogless has a deficiency on frogs. I call it a frogopenia. So an ecosystem can be sick, and one of those sickness can be the lack of different kinds of animals. And the frogs being a very important connection point within those ecosystem networks, whatever they are inserted into, whenever they lack into the ecosystem, they unbalance that ecosystem so much that the whole ecosystem can literally go into collapse. And if an ecosystem is collapsing, also the society around it will start to collapse too, because most of the resources that they might look for might depend on that ecosystem where they're near to. So everything is connected. The balance within the ecosystem, the balance within human health and the society health as a general, and also the living beings and the health and balance of those living beings living in that area. And a veterinarian happens to be a very key professional on it because addressing problems within animal health, it can prevent those animals from disappearing on the ecosystem network, therefore keeping the balance within that ecosystem. Also, we can prevent literally health issues, especially zoonotic issues, of spreading up like as a spillover or spillback from human society. So being in this middle field make us a very important source of information, source of knowledge to help the One Health approach to help to happen and to be sustained. And most of the time, we try to avoid it, or we don't even know that it's possible for us to be involved within it. Well, then the amphibians are a practical example of it. So, if you guys can picture, the amphibians were in Central America during the 70s. They were having a lot of trouble. We in Central America, we started getting like markets looking for supplies for European food chains. And one of those supplies were frog meat, because in different places of Europe, frog meat is very much appreciated, just such as in France, they eat the frog legs as one of their main dishes. And the tropical environment in Central America was very good for them to try to breed these amphibians and sell them to these food chains. So having a big potential for culture, which is the frog breeding, helped the the frog help that those places start to develop a lot of frog productions for meat, because big demand will be aligned with big profits, and those people that were trying to get their lives back in Central America, especially different producers, individual producers, they were trying to look for a niche for them to fill up. They found into the raniculture a big opportunity for growing. By then, the native frogs were just like chilling, frogging in the jungle, not worried at all about what could happen in the future. But then to fill up those rainy culture, the Central American frogs, they were not good for it because they were quite venomous and they don't have much meat. So they had to import different frogs from other parts of the world. And one of those species that were very good for frog meat production were the bullfrogs. So the bullfrogs got imported for the raniculture purposes, and they adapted very well. Bullfrogs are great for raniculture. They have like a very large uh, weight conversion. Most of the body can be sell after the animal has been has been killed, and it's very accepted with very good quality for the restaurant and food chains demand. So they were imported, and they started growing it in Central America. What could go wrong, right? But then some things went wrong. Some of the problems that started to begin were that nobody predicted that the bullfrog production didn't have the best biosecurity measures. So bullfrog, the waste of the bullfrogs, that water that has been used was being discarded right into the natural sources. So there were like a water stream, a natural water stream, and those wastes from the bullfrog production, the water waste was being released right by the natural places. So native frogs start getting worried. Some other things start happening too. There were some storms, hurricanes that destroyed many rainy cultures. And all those frogs from those rainy cultures, some of them died, some of them got into the wild because they literally got their outbreak. They got released into the jungle. And they loved it because, I mean, with tropical environments, it was very easy to adjust. And special for some species, they are so adaptable, such as the bullfrogs. So they got introduced, and the native frogs start getting worried a lot. And that's because the bullfrogs, they literally compete with the nature frogs for their food source, but they also eat the native frogs. They are also resistant carriers for the chytrid fungus we've mentioned before. A matter of fact, uh, at that time in the 70s, when all of this started happening, the bullfrogs were the only ones that had their skin uh, histologically studied. And in those histology studies, they've already seen that there were some cysts into their skin, but they didn't know what that was. And since the animals were healthy, they didn't thought it was a problem. So when this fungus started contaminating the animals in Central America, some scientists started collecting skin from frogs in the wild, but then they would use as a reference those works with the bullfrogs. So whenever they identified that there were the cysts of the chytrid fungus, they thought it was a natural thing because the bullfrogs had it and they didn't have any sign of disease. So no one knows that the fungus existed at the time, although they saw the structures in the histological cuts and in research, they thought it was part of the animal. So, as no one known the fungus exists, also not many people cared about frogs or ecology in general, even less for frog ecology. There were not many people that knew about frog diseases, and there were not many people that would care for the die-outs other than the scientists in itself. It took a long time for scientists to understand what the problem is. Actually, the frogs that were started to get worried a lot, they were literally doomed. I mean, that fungus carried by the bullfrogs, it spread it so fast and it reached so many places, so many different species. Those frogs eventually got extinct. And the fungus, it literally decimated the Central American frogs. And it took 20 years. It was only by the end of the 90s that the scientists finally discovered what the problem was and that it was a fungus that was causing the trouble, especially because the chytrid fungus had never caused problem into any animals before. And it was just the frogs that they started getting like a personal thing with them. But 70% of the amphibian species in Central America got extinct in those 20 years. 70% loss on richness, on its species amount within the same place with animals from the same class is the hugest mass extinction in a short amount of time ever registered in the world's history. It was the biggest die out of a same class in a shorter amount of time ever seen. So this is compared as being way worse than the asteroid that hit the Earth and killed the dinosaurs. So this is the biggest wave of extinction in such a short amount of time for the same class of animals within the vertebrates. And nobody seems to be talking about it. And you know what caused it? A disease. So those animals were sick. Who are the ones responsible to care for frogs' health? Us, veterinarians. And what did we do? We took 20 years to be involved until we decipher what was happening. And that delay caused the extinction of 70% of different species of amphibians. Imagine the impact of that into the One Health. I will show you. This is the impact on One Health. This graphic shows us the increase in the amount of cases of dengue fever into countries of South Central America in 40 years up to now. And do you think that the spread of dengue might have something to do with the enormous decrease on the population of the predators of the mosquitoes that are a vector for the dengue fever? It looks like it's important, right, for us to address this problem. And if veterinarians had taken action before, well, and given the relevance to the case that it deserved before, we might even have prevented die-outs in people because of spread of diseases. So health is all connected, and veterinarian is a key professional into preventing it from happening. Not only helping it to decrease the amount whenever it's happening, but only to prevent it from happen, most importantly. So learning about frog health looks important, right? So we only we not only can look for the frogs that are sick and getting into our clinics, because that's going to happen even more and more often, because frogs are spreading out through internet as pets, even though they make good pets. The people will buy them because there's people selling them. But we also, as a veterinarian, should be within the ranicultures to make sure biosecurity measures are being taken care of to prevent spread of diseases into the natural sources that their waste might be going to, but also to take very good care of the animals they are being produced in there, because literally the ranicultures I've been to, the frogs they are being like raised in there, they are in awful conditions. I mean, they most of the times don't have veterinarian care at all. And if it's just a discard animal if it gets sick, and the health is not even like controlled as a whole group. So if one animal is sick, they discard a sick animal. They don't even try to check on the health of the other ones. So the we have to make sure any cultures uh, get a better veterinarian care, and that can also impact the human health too. We also have animals that are being kept in zoos and other places for conservational purposes or for educational purposes that might get into our need of assistance within veterinary care. And there's also, this beautiful red-eyed tree frog. This is a this is Brazilian's most endangered species. This frog lives within an area of 100 meters square, 10 by 10, within three masses of bamboo plants and with a little stream passing in the middle of them. And that's the only place it's found on Earth. We are aware of 16 of them living in that place. And now that it has an ex situ management too, it's been holding to a zoo for research purposes and for conservational purposes too, because the species is literally on the brink of extinction. And it's one of those species I told you guys that if a cat reaches this bamboo where this frogs lives and decides to kill the 16 frogs there and there, like within 2 weeks, eating one every day, this species is extinct. This is the amount of threat that these animals are being put through. And, you know, imagine if Kitrid hits him. It hasn't, but what if? So many other species are having the same situation as this one. And it might be happening to species in whatever country you guys are too. And us as a veterinarian, we have a responsibility to take care of these animals. We can. We can not only help to prevent the disease spreading, but we can also monitor the disease spreading, monitor the health of the frogs in our care and into the nature. We have so much potential to help these animals. We need to take action. That's mainly the most important message. And that take us to a sick frog. Okay, so now I have a sick frog. What do I do? That's like a very good question. Most people will say call Iago and they will send the patient my way. And I'm good with that. I love to help, and it's always good that people look for me for this sort of information. And I'm glad I can use whatever I've learned through the years to help these patients. But I am only one person, and there is so many frogs needing assistance that I started doing this, like trying to help as many as I could. But I figured that I cannot take all the frogs that are sick all around the world with all the help that they might need. So I need to put more people out there trying to help frogs too. And that's what we're doing here. We're trying to spread the word. And I hope you guys feel like impacted by the amount of need that the frogs might need and get influenced into learning more about amphibian health so you can help the amphibians, at least the ones near where you are. Well, I'll show you some on what to do, at least for you to know where to start, okay? So one thing that we do is that we use and abuse from the semiology knowledge. We have to add to that semiology some common sense, do a little bit of research, ask for help from people that know what they're doing, just to check that if you're doing things right, just to get like little retouches on how to do things better. But the semiology will take you a long way. And to start semiology, we usually start with the anamnesis. So we do a review of our patient. Who is our patient? It's very important for us to take note on that because even though within the same group of frogs, like the Anurans, there are like a whole order, we have so many different species with different sizes, different ecologies, different anatomies, different physiologies. These three frogs, for example, the ones I'm circling with my mouse here, those are philomedusas. And philomedusas, they have different management of water loss as they cover themselves in serum instead of covering them in mucus. And this serum part, it protects them from dehydration way better than the mucus does. To also avoid the lack of water, they, instead of excrete the urea, which might need water to be excreted, they convert the urea into uric acid and excrete the uratus that we usually see in in reptiles and birds. So most of the frogs don't do that. Most of them will do urea if they are terrestrial, or they will lose the ammonia to the water if they are immersed into the water. So we have to know which animal are we used to work with, because at least if I seen an animal that's dehydrated, it might have different causes of that dehydration. It might be the skin the problem, it might be the kidneys the problem. So we have to understand their physiology in order to understand what kinds of procedures and diagnostics should we run. There is also a lot of things that we do in the anamnesis, like trying to find a history for that animal. If the animal is like a wild animal, it might not have a lot of history, but at least like how long has the animal been with you? And if the guy or the person who's bringing the animal knew or saw what happened to the frog, it might lead me to a better way of handling the problems or the lesions that the animal has had. The if the animal is a pet animal or a zoo animal, or if it's under animal care for a short amount of time, it might have a history. We might know like for how long it's been there, how long the problem started, and the kind of problem are usually different too from the ones that wild animals usually get. Wild animals are usually animals with lesions either by uh, problems with humans, people that are scared of them and hit at them with something, or they got caught by a pet, just like a dog or a cat, and they might have trauma because of the bite, for example. Or they are road animals that got like hit by a car or something like that. They might get trapped into different places of the house and might hurt themselves. They can also fall into places where they weren't supposed to, just like a pool. We know that to clean a pool, we use a lot of chlorine, and chlorine is very highly toxic, not only for the frog skin, but for the frog bacteria in the skin too. And they can cause dermatitis in itself, but it can also cause the skin dysbiosis that we talked about in the next days. So it might need veterinarian care for a couple of days before it being okay. So knowing the history of the animal will help us a lot on trying to navigate what kind of the problem is and what might be the right approach for them. To if it's an animal kept under human care, then we might know what's the origin of this animal, if it's from the traffic or not, what how it's the environment where it's kept, what are habits that the animal usually shows, how is the feeding, how's the management, how's the cleaning of the enclosure. So all of those are questions that we might need to obtain. One thing that's very much important is to not only collect information about the animal, but also about the environment where that animal has been inserted into. So, if the animal is an aquatic animal, get a sample of that water, run tests on the water, because the problem might be the water and not on the animal in itself. Also, does the person that's kept keeping this animal keep a record on humidity? Does it keep a record on temperature? Those are informations that are very important to have too, because fluctuations, especially very hard fluctuations on temperature and humidity, might cause a lot of problems. Cold waves or heat waves are very degrading to amphibians' health. If an animal has a very small tolerance for heat, for example, axolotls, the salamanders, you might know what I'm talking about, right? I hope so. They are usually kept in cold water. So they are used to water around like 15 degrees Celsius to 18 degrees Celsius in max. If it's a little bit over 18 to 19 or 20, they have a lot of thermal stress. They can even die of thermal stress. And the water, if it doesn't have a very strict control on their temperature, and it might need a chiller to keep the water cool, if those fail, the water temperature can raise and kill your animal like in one night. So if you're having an animal that needs that sort of specific environment and environmental conditions to live, you have to be prepared to offer it at all times. It cannot fail because failing means the animal might die too. So they are this sensible. And whenever I see people selling those animals like the axolotls um, in pet stores, in aquariums and fish stores, I always think to myself like what the hell, you know? I'm sorry for language, but um, axolotls are critically endangered species from Mexico. And instead of us taking action to try to keep those animals alive and then their environmental conditions in their environment there in Mexico, what we do is that we are spreading different kinds of lineages of axolotls selected by humans to have like different colors, just like animals that are completely white or albino, uh, those that are a little cystic, and we're using them as pets while the species is struggling to survive. Where is it from? And having those animals in under our care, it doesn't help conservation in any sort, especially because the animals we're getting, they have terrible genetics, and buying one axolotl doesn't help conservation at all, because the money doesn't go there. Money goes to places they are reproducing them, like in whatever. It's not the point, but it just gets me like a little bit angry that we're not focusing on getting those animals to survive in the wild sometimes. Well, apart from anamnesis, we go for inspection. And inspection is done hands free. We do not touch the animal to inspect it. And as we inspect the animal for its pattern of activity, their locomotion, their body score, if they're too thin and they might be showing like some of the bone lines. We also check if they have an individual ID that might be a pattern of the skin or a microchip or something like that. We check about their color. This animal, for example, I showed you guys the Philomedusa here. It's dehydrated, and we can tell it's dehydrated because it's usually very greenish and with the skin that has at least the shine of moist. Whenever it doesn't have the shine of moist, whenever the skin has changed the color to patterns that we might identify as a problematic pattern or a dehydration pattern, such as this kind of color for green animals, we can, like, just looking at it, know for sure that's being dehydrated, and we might try to look for a cause whenever we go for the next step, which is the physical examination. So just so you guys understand this change of color. Animals within their skin, they might produce three kinds of chromatophores. The amphibians, for example, do just like three kinds of chromatophores. One of these chromatophores is called melanophore, which is the most basal one, the one that's the deepest, and it produces melanin. Melanin is usually black. If it's conjugated into iron, it can become red. So animals that are red or black, they have just this increased amount of the melanophores, making them look that color. For the animals, they are yellowish. They have centenophores. The centenophores, they usually produce colors and pigments using carotenoids that the animal gets from their food. And these carotenoids, they usually make the animal yellowish to orangish. And then there is another layer called the iridophores. And the iridophores, they don't usually produce a pigment, but they make it a crystal. And this crystal can reflect and deflect light. And the color that it usually reflects is the color blue. So whenever we see in a blue animal, that's not a true color, that's an optical illusion based on the reflexing of the blue color out of those iridophores in the skin of the animal. So when the animal have layers of multiple colors, they can get different colors out of the blue, the yellow, and the black or red. So this example is a Philomedusa. Philomedusa, they have a small amount of melanophores, and then they have a lot of centinophores and a lot of iridophores. And those layers of iridophores put it into the top of those of centinophores, mix the blue with the yellow and make it the color green. And there is a thickness to that skin that whenever the animal is dehydrated, it makes the skin even thinner, connecting the melanophore to the centinophore and bringing the iridophores so close to the centinophores that the iridophore reflection cannot be perceived. And then the mix between the centinophores and the melanophores resultant to that makes it the color brownish that we see here. So this is a dehydration color because we know here that this skin, because it's not moist, because it's dehydrated, it's thinner and the iridophores are not reflecting the blue. So we're just seeing the resultant colors under it. And that might happen to different species too. But for example, if we have a blue animal that's dehydrated and their skin gets like too thin, it might just like lose the some shades of the blue color and become darker. Not always color change mean dehydration. It might mean different things, but dehydration might be on our list of differentials. Okay, so there is one thing that another thing that we look for in inspection is the respiratory rate. We usually do this with the animal without being restrained because as soon as we restrain it, it might increase the movements, the gular movements that we call the respiratory movements. And as it doesn't have ribs, what we do is that we cannot count the respiration through their laterals or their flanks. We have to look for this area here, the gular region, because that's involved with the active respiration. Amphibians have three ways of respiration once they are adults. One of them is the cutaneous respiration, the skin respiration that we've already talked about. Another one is that they have a oropharyngeal respiration where they fill up their mouths and oropharynx with air, and as they compress it, it changes the pressure within inside their mouths and the gas that it's in there, and it forces the gas to make a better exchange of rate of the oxygen. And once they compress it, they usually also open up their trachea for the air to pass in through it into the lungs, and that's how the lung get inflated. Once they open up their pharynx and their trachea, the air comes out to the mouth again, and then it liberates the nares, and the air goes out. So the air makes two oxygen exchanges, one when it's inspirated and it stays inside the mouth, one when the mouth comprimes the cavity and the oral cavity and sends the gas into the lungs, and another when the lungs come out, let the gas out, and it comes out by the nares. So when we not begin with the anamnesis, usually when it's an emergency. So what are emergencies of frogs? We usually in veterinary medicine, I don't know about different parts of the world, but here in Brazil, we learned that there is a ABC of emergency, in which the A stands for airways, B for breathing, and C for circulation. So we check perimeters in this order. We first of all we check the airways and check if it's not like obstructed or broken or something like that. Then B, we check the breathing, if the animal is capable of keeping their respiratory movements. And C, we check circulation, and we check then the pulse, the color of the mucosa, and if there's hemorrhages. And we try to stabilize those parameters in order of importance before taking care of different kinds of emergencies that we might have in different parts of the body. For amphibians, their airways might be obstructed, like the nares and, and the larynx, but the skin will persevere. The skin will be able to keep the oxygen exchange, and in that I mean that even if the main airway for the lungs are obstructed, these animals can still breathe by other means, which is the skin. So the airway it's not important anymore, or not that it's not important, but it's not as important as it was in other animals, where it's the main place for the air to pass to be exchanged. So it loses importance and it goes down in the list. And then we have to bring the first one to be the skin. So instead of a ABC, we have a SBC, which means the skin, then breathing, then circulation. Because checking out the skin, we make sure that even if the airway is impacted, the animal will be able to make oxygen exchange. And then we check breathing with the main airways for the lungs in itself, because that's the only breathing we can evaluate, because we cannot evaluate if the skin is breathing or not. We can just infer that, because the skin, if it's moist and if it's not inflamed, it might be working. So, or we might get different signs from it, or at least a sign that it's not working if the animal starts to having their mucosas a little bit bluish. And then we check for circulation, just like we would in an animal that's passed through the ABC uh, emergency management. Then, what is an emergency in amphibians? Especially cutaneous problems. Extreme dehydrations will be an emergency. Extensive cutaneous trauma is an emergency. Hemorrhages are emergencies too. Commuted, comminuted fractures, completely problematic in amphibians. And for fractures in itself, it's hard because the skin of amphibians does not allow any bandages. So if we put a bandage through the skin, like an immobilization bandage or a curative, it it will make the skin goes into necrosis. So we cannot place anything in the top of the amphibian skin. We cannot make a bandage. We cannot make immobilization. So to do the treatment for the fractures, we usually do surgery, which requires internal fixation within the bones. And then the openings where those fixators will be passing through will be open in, will be wide and open. And we will have to take care of them as open wounds without placing any sort of like a gaze or anything like a tissues or bandages or anything around it. So it's quite challenging for us. Intoxications can be also emergencies, especially when the intoxication is for organophosphorates and carbamates, which are poisons used to kill some of the amphibian sprays, and the amphibian can prey on intoxicated prey and get intoxicated also. Another intoxication is when it falls into the pool, as I told you guys, or when people um, as uh, people that has nothing to do and mean people put salt on it. So it's not uncommon for us to see that people are scared of frogs or they don't want they're near them, and they make salt traps, or they throw salt into the frog in order to try to kill it. And those frogs are usually very dehydrated and have severe skin lesions because of the salt when it comes for us in a clinic. So if it's not an emergency, or if it is an emergency and we need to do a physical exam, how do we do it? We always use gloves, always use gloves, and always powder-free gloves. Oh, I'm sorry, I forgot to translate this, but I'll I'll tell you guys what is written in here. So here in the top it says always use powder-free gloves. So gloves are not only enough, but you have to use the powder-free gloves, because the gloves that have the powder in it, that powder is also toxic for amphibian skin. And then what material is best? We usually have latex gloves as the more common gloves that we have out there. They're not bad, but they have a little bit of um, how's it called? They are not as smooth, so they can, if you're managing it with a dry latex glove, it might still have some friction into the amphibian skin, and we might not see like a toxic aspect of it, or a trauma, but it can make the amphibian skin usually more reddened. And then there's also the vinyl gloves and acrylical gloves. Those are also good, and they have way less friction than the latex one. We can use all of them if they're powder free. Okay. Oh, Yago, I found a frog in my house and I don't have gloves here, but I want to remove it. It's not sick, but I have to get it out. How do I do it without the gloves? Well, here in Brazil, it's very common for us to have those plastic bags that you get in the market. So if you have one at home, you can put the plastic bag on your hand, put it under the water on your sink, let it wash and get very soaked, and then use those plastic bags to do the handling, not doing the handling on your bare hand, okay? Because that it's very bad. So to hold the frog, it can be done in any frogs. Okay, so any frog or any amphibian in general can be held, and we usually do the right handling, making it be held by the inguinal region. So this region here in their waist is where we place our hands, and we use our fingers to keep the legs spread, like completely spread out, so it loses the ability of trying to jump and get to push your hand. Okay, so here if the animal is too big, you have to support it with a second place, but if the animal is tiny, you just have to hold it by the waist. And we usually do like we put our top, like our fingers together, just like a water drop, and we hold their waist by the top of our fingers. We always check from nose to toes, and there's some things that we're usually looking for, just like asymmetric characters. We look for lesions, changes of color, especially on places that are like skin folds. um, Always look for the feet and under the feet. Animals that are being kept in bad substrates, they might get pododermatitis. But some animals that have infectious diseases, and some of them are quite bad, might also have lesions on their feet. So BD, that fungus, the chytrid fungus, might cause lesions in the fingers. So that's a place where it's usually very common for them to show up. But also another one called mycobacteriosis, the same genus of bacteria that causes tuberculosis in humans. It might also contaminate frogs, and they might cause two kinds of disease. One kind of disease is when they make the granulomas, just like tuberculosis does the granulomas, but it can also cause the skin lesions where they open big wounds that do not heal. So those wounds usually open up very commonly on the feet. So whenever you have an animal that has lesions on the feet, we always test it, even though if it's like very supportive that it is pododermatitis because the enclosure doesn't have uh, the right substract, we always test it just to be sure, especially because those mycobacteriosis, they can be zoonotic and cause us trouble too. Um, we test for cutaneous turgor, so we usually pull up a little bit of the skin folds and see how long it takes to go back to the place. We, if we want to, and not if you want to, we have to. We open their mouths, and to open their mouths, we have to remember that their mandibles is just like arch of very thin bone, and to open their mouths, we usually use a card in the commissure of the mouth. And as soon as it starts to open up, we put our fingers in both commissures and we hold it open by the sides. Never keep it open by the top of the mouth. Okay. And inside the mouth, we usually see those very two big masses in their palate. These big masses are their eyes, and their eyes and mouth don't have a bone structure to separate them, so they only have mucosas. And these skins that separate the eyes and the mouth are this way because the eyes can be pushed into the head and they help these frogs to swallow their food. So they are used for mechanical purposes also and not only for vision. Here in the top, we also have the aperture for the nares. Sometimes they have this kind of formations. They are called the vomeral teeth. They're not real teeth, but they're bone projections that help to trap their prey into their mouths and also to squash their prey when they're inside their mouths. So the tongues for most of the amphibians and the neurons in itself, it will be folded back into their mouth. There are some frogs that are tongueless, just like the Xenopus genus, the long-clawed frog that's from Africa. But most of them will have the tongue and it will be folded back. So we have to use an instrument to fold it forward because we have to see the top surface of the tongue. That's a place where the rhinovirus lesions usually show up as hemorrhagic spots. So we always try to check that. We check the colors of the mucosa. We're looking for a pinkish tone, just like this one. The tongue is usually whitish. Okay, so do not confuse the animal for being anemic or something like that because the tongue is pallid. Use the color of the palate as your reference. Here in the sides, we have Eustachian tubes that connects to their inner ear. Behind the tongue, we have the aperture for the pharynx and the larynx and the esophagus. And if it's a male, we pull the tongue to the sides, and on the sides of the tongue there might be another aperture too. That's for the vocal sacs. And so for animals that we cannot distinguish if they're male or female, opening their mouths and checking if they have those vocal apertures for their vocal sacs, it's a good way for us to sex them. We always check their ventral parts too. And if you pay close attention, there is an area here within the inguinal and ventral inguinal area, and also those tides that have a very glabrous skin. You can see the skin is different. It's not uh, as as smooth as the other parts. This area is called a drinking patch, and in this area is where most of the osmotic regulating cells stay. So this is the place where they get the most absorption of water, and also they can also lose water through here in the solutes such as ammonia. Um, so this area is very important because if there is a lesion, even that's like a small lesion in this area, the whole animal can get dehydrated because this region is very important for us. Here I have an example of it. It was the first philomedusine that I showed you in that changed the color to brownish. Here it has an inguinal lesion. It got stuck into a place where the front portion of the body passed through, but the back wasn't being able to, and it got trapped. And this long lesion here caused it to be dehydrated for a long time, and we had to use like further baths to rehydrate it constantly. So as we check it out, like the whole body completely from the outside. Oh, I didn't tell you, but if we need to check the vitals, we for respiratory rate, we count the gular movements that we did in the inspection. But for the heart rate, we usually place a Doppler between the insertion of both arms where the heart is, and we get like a very good listen of the heart. The stethoscopes don't work well in these animals, so they although they might be used for some, especially if they're too little, you won't listen to anything. So use the Doppler for every species of amphibians because that will always be better choice. Okay. So for pulse, if we need a pulse, we can try to use a femoral pulse as a reference. They also have a pulse that we can try to find. And for temperature, we do not take cloacal temperatures. We just measure the temperature that's in the room because that will be equivalent to what the animal is. And for complementary exams, we might do a blood draw and run a hemogram and biochemistry. For a blood draw, we usually use this ventral median vein that the frog has very superficially and the median line of their body. It's the main site for us to collect, but if the animal is too small, we might try to use different approaches. The imaging is very resourceful too. We can use X-rays, especially to check on lung problems and bone problems. But other than that, it can be quite tricky to evaluate other organs by the X-ray. So if you have like a metabolic disease, which is quite common on our clinical cases, the X-ray is like the gold standard for us to checking that. I've even had animals that had worms in their lungs, which are quite common too, that we can see the worms inside the lungs with X-ray. I've had animals that had torsion of the lungs, so we can see the silhouette of the lungs are asymmetrical, or might even have like a line, like looking like they've been torsioned. So it's quite useful for us on some diagnostics. And the ultrasound is also not a good option. Whenever I do an ultrasound, I try to put the animal into a plastic bag or a plastic container, and I fill it up with some water or some of the fluids that it might be bathing on to rehydrate or to dehydrate on purpose. For example, this one is a pepper frog, also called the chicken frog here in Brazil because people say that it tastes like chicken. That it had anasarca, and I was trying to find like what's the source of the anasarca. So I decided to make an ultrasound to check the liver, and eventually I decided also to check the heart. And when I did check the heart, I found that the animal had oh, say it in English. It is a cardiac. So I'm thinking about the name. Sorry, guys. I English is not my first language, so I'm trying my best. Okay, so it had a heart disease where the blood accumulates within the ventriculum and the atria because it has insufficient closure of their valves. So I think it's congestive cardiac um, insufficiency. I think that would be the translation. And then for the microbiology purposes, we usually do swabbing. And to perform a good swabbing, we usually pass the swabs first place, always the drinking patch, where I just told you guys where it is. And then we got the cloacal and back of their thighs. And in the end, we pass it through the top of their fingers and their palms or their plants of the feet. And if the animal has a lesion that's an open wound, we leave it for last, okay? Because it probably has a lot of bacteria and we don't want to spread that to other places of their body. And with this kind of swabbing, we usually do 10 passing of this swabbing in every place. And this swab, we can put it into a little container with saline and centrifuge it, get a pipette to get the sediment of the content and make smears out of it. And with those smears, we can stain it to look for gram bacteria, so a gram stain. So we can check what's the relation between gram negative and gram positive bacteria. And although there's some species that might have different percentages of each skin, usually health skin do not have over 30% of gram negative. It will be mostly gram positive. And if it's like largely gram negative, it's probably what's causing the dermatitis in itself. We can also do a right stain, especially to look for fungal. So fungal IFs or for fungal, I don't know names in English, I'm sorry, but those formations that look like a footprint. And we can do also the alcohol acid fast stain. For those fast stain, we are looking for bacteria that do not color with grain stain. And those bacteria are usually the mycobacteria that I told you about, like the wounds that won't heal, especially on the foot. So we can try to look for them in the acid fast stain, and they will show up most of the time. It's not always guaranteed that they will show up, but if they do show up, we can conclude that it's an acid fast and most probably a mycobacteria. Unfortunately, whenever we have a positive of mycobacteria, we usually recommend euthanizing the animal because we do not use our medicine that we use for human health to treat mycobacteriosis in animals, at least not here in Brazil. So we don't have a way to treat them, and as they are multi-resistant, we just unfortunately have no choice other to send the frog to the lake of the Lord. And well, that same swabbing can be used to do PCRs too. And whenever we have like a suspicion of the BD, for example, that's the swabbing we do to send for a PCR evaluation. Well, and what if I need to medicate? This is my last slide, I promise. I'm finishing. So if we need to medicate, there's some sort of options that we can use. One of them is the topical medication. And topical medication is very, very, very useful in frogs. So most of most of the different drugs we have can be absorbed through the skin due to the skin permeability. So if we want to do fluid therapy is done by immersing the animal into a liquid that's isotonic to their body. So if you guys uh, want to know as the commercial fluids, the NaCl, the saline at 0.09% is usually hypertonic for frogs, so it causes dehydration, so we do not use it. Common ringer, also hypertonic for frogs. It causes dehydration, we do not use it. Glycosis at 5%. It's very hypertonic for frogs. We only use it in cases, for example, where we had that frog with anasarca and we want to reduce edema. Then we use the hypertonic fluids. But then if we're using it, the glycosis 5% is very useful in that matter to reduce edema in itself. The lactate ringer, we don't use that, it's completely forbidden. The frog will absorb the lactate, and this lactate won't be uh, excreted by their livers, just like mammals or birds would. So it will accumulate, and those lactate will be converted into lactic acid and will cause acidosis into the frog. So we do not use it at all. So what should we use then, right? I mean, we're running out of options. For frogs, we can either manipulate the like a compound the formula in a pharmacy. We send them the recipe with the minerals we want in it and ask them for diluted in sterile water, and they will send it to us. That's the best isotonic fluid that you will get. And as it doesn't need to be sterile because it doesn't go into the veins, it goes into a little pool where I'm going to put the animal in. It's very amazing. I mean, you see frogs literally resuscitate after being like very dehydrated and being put into the water. And the best part of it is that we don't need to use like a catheter. We don't need to get a vein in it. We don't need to regulate the dropping and nothing of that. We just put it to rest into a little pool for a while. And conditions to do it is that the skin is full. The skin cannot be inflamed, otherwise it won't absorb. Then we use the parenteral via to rehydrate it. But we usually don't get the veins. What we usually do is that we'll do intracellular injections of those fluids, but it's quite rare, okay? But for most part, the bath will be like the best way to get the animal to be rehydrated. What if I don't have that compounding pharmacy, or if I don't have it right now and I need it like right away? What you can do is also get like a mineral water out of like a bottle that you bought on the market, and that water will be more isotonic to the frog than any of the fluids that you have home or at the clinic. So try to use that too. And if you're in the wild and you find a frog that's dehydrated, you can look for a water fountain anywhere near that place where that frog used to live and do the same procedure and put them into those little containers into the water, and the frog will rehydrate very well. Okay? So when do I use parenteral vias as well? So I already said that I use it when the animal has like skin lesions and I need to do fluid therapy, and then I use the intracelomic way. But I also do injections whenever I have to do like very specific dosage of a medicine. If I have, for example, anti-inflammatory, just like an NSAID, meloxicam, or ketoprofen or ibuprofen, and I want to use that in a frog as an anti-inflammatory, I can literally calculate the dosage and put it into the top of the head of of the frog, like in the skin, it will absorb and will take effect. You do not need to poke it, no need for injection. It will absorb it from their skin. But the amount that's fully absorbed, we cannot control. So if we need a very strict dosage, just like antibiotics, then I prefer to do it through injection because I know exactly the amount that's going to be injected. And I have more control of it. And then that's when I use an injection for. When I do injections, I have to remember that frogs, just like reptiles and birds, they have a portal renal system. So if I do injections in their legs or in their caudal part of the body or intracelomic, they will pass through the kidneys and might be excreted before getting into circulation. So we have to use the arms and the muscles here and the shoulders for injections as the best points that won't pass through the portal renal system. Okay? And Antero, I might use it, but it's quite stressful for the frog. So I usually use Antero, which is like orally, giving it the medicines orally. For frogs I want to deworm, and I use like the deworming medication that acts topically within their inside of their gastrointestinal system, just like fenbendazole is one that I use a lot. Or when I need to give like uh, supplementation for them, just like minerals or even like artificial feeding or tube feeding for them when they're not eating. So to the last topic I have there is anesthesia. Anesthesia can be a little bit tricky in its training for people to do it, but some things change from what we are used to other animals. Whenever we have a frog, we have to remember that their skin is the main part of the respiratory system. So if you want to use the parenteral medication, just like ketamine or the alpha 2 agonists or any of those, they take a long time to metabolize in the frog's body, and the frog will take a long time to wake up, especially if you don't have the reversers. So what I do most of the time, as I don't have the access to an MS222 here in Brazil, which might be available wherever you are, or I don't have alfoxylone, which is also another one that's quite used for amphibians. Here in Brazil, it's forbidden. I use a lot of isoflurane. So either isoflurane or sevoflurane, which are inhalatory anesthetics, can be very good for frogs. We have to pay attention to some things. Whenever we want to induce anesthesia, we place them into a container, or for example, here for the tiny little frog, I use the mask as a container for them, and I regulate like the inhalatory anesthetic into it, always monitoring the heartbeats and the respiratory movements of them. Heartbeats, I monitor it by placing the Doppler and putting the Doppler like stuck into a place I want to keep it. And then I put the frog on top of it because sometimes the frog is smaller than the Doppler, but the Doppler still works. Um, and then the respiratory rates, whenever I have a animal that stopped breathing by their lungs, I know it's getting into uh, a good anesthetic protocol. It usually does uh, the it usually stops breathing within the anesthesia. So it's expected from them. But we have to remember that the skin's still breathing. So the monitoring now, as soon as they stop breathing from their lungs, is that opening the mouth to check on the color of the mucosa and see if the skin is doing the work of doing the gas exchange and not getting it cyanotic. We also check the reflexes. One of the reflexes is the eye reflex, like closing the eyes, and the palpebral reflex. But some other reflex that we also try to pay attention to is that the positioning reflex of the legs. And whenever they are already into a more deep state of anesthesia, we flip them into their back, and we see if it has like the reflex to come back into the orthostatic position. So that's how we try to monitor them while they're anesthesiated. Sometimes you'll see people also mixing the isoflurane with gel, like a water-based gel, and uh, sterile water all together to make like a fluider gel, and we'll put drops into the amphibian body, and the amphibian can also inhale the isoflurane from this liquid. It's a little bit harder to dose, and we can do like overdoses of this anesthetic that way. So I use it sometimes, but I try to avoid it as much as I can. Um, Another thing we have to remember is that whenever we are anesthetizing it, the gas usually makes the skin drier, so we have to keep wetting the skin, because a skin that's dry won't do the gas exchange. So we have to keep the animal wet. So I keep like putting drops of sterile water or their fluids into them, like during the whole procedure. Usually like every 2 minutes or every 5 minutes in the max. You see, there's also a bag under the animal. I usually heat up this bag, not enough to burn the animal or whatever. It probably is around like 26, 25 degrees, and it will keep the animal also heated up, at least at a higher temperature than the room will be, unless the room is like really hot, but that's usually very bad for the surgeon or for the one doing the anesthesia. So whenever I can, I try to control the temperature at least close to where the frog is, to try to keep them warmer too. If you want to, you can even try to get the water that you're gonna be dropping on the top of it a little bit warmer too, instead of like a very cold water that will like keep the body cool. So those are ways for you to try to get that the medications going. And if you need to do analgesia, there's some medicines that we often associate to create a very good anesthesia, a very good analgesia protocol. One of them is the NSAIDs, like the within the anti-inflammatories. I try to avoid any kind of um, steroidal anti-inflammatories, so I just use the non-steroidal in them. But another one that's quite useful is, here we call it dipyrone. It's a COX-3 anti-inflammatory, and it's very good for them with a long-term analgesia. And if we need, we can associate an opioid. But remember that frogs, they do not have the me receptors of opioids. So methadone, morphine, tramadol, none of those work. Actually, tramadol doesn't work at all. Methadone need huge doses for it to work. For example, dosage of 90 to 100, 150 milligrams per kilo for it to work. So it's just like ridiculous. We don't use it. And the one that works very well are those that have kappa or beta receptors. Um, Budorphanol is one that's very good. It also causes a little bit of sedation. So if we have like a frog with a fracture and we want to keep it like less active, moving less, just to help it to keep the position of the the immobilization of the leg and all of that. and with an analgesia effect in the top of it. We use butorphanol in those cases too. So it's always a combination of drugs. You see, guys, that I didn't put any dosage here. I'm not sure if everybody that's watching or will be watching is a veterinarian. So I didn't want to pass any kinds of like strict protocols with dosages in here. And in addition, it's not all the things that we do that are here. So there is a lot of layers and more knowledge to be transferred into amphibian medicine in addition to what you guys learned here. So I invite you to keep learning it from me, from us, our team in the future too, if you want to know more about amphibian medicine. This was a very long introduction to what amphibian medicine is and can be. And I bet that the sick amphibians that you guys are going to be um, are be caring for will be very happy and will thank you for looking for this knowledge to care for them. And remember, this is only an introduction. There's so much more to learn. And if you need me, you can reach us out. I can be your focal point of assistance, and I can also help you with further training if you want to, and if you need to, and if you have frogs that need you. Okay. Um, if you guys want to, there is a Portuguese course available on amphibian medicine with me on our website, www.wildplace.com.br. But although it has English subtitles, it's fully online, and you're gonna listen to me like speaking in English, so you have to like to learn to read the subtitles to get it. But another option is to um, with the ASG group, we are planning on starting a veterinarian task force for amphibians around the world. And if you want to set up in-person training wherever you are, please reach out because we can plan on that. And then I can go to wherever you are to give you the training and to help you understand the needs that you might be getting, whatever you're keeping these animals or seeing them in the wild. And, you know, ask for help. We're here to help, and we want to make the difference for the frogs all around the world, not only in Brazil. And we want to help you to make the difference for the frogs that are near to you and that might be needing veterinarian assistance, just like those that I showed the examples here. I'll speak in Portuguese really quick just to call Victoria because she's gonna put the keyword for you guys here. Vitória, você coloca a palavra-chave para nós? Then, with this keyword that will show up on the screen, you go to our forms and you fill it up if you want to get a certificate for this lecture. If you don't need a certificate, don't do anything, okay? Just follow us on social media. We'll be happy with that. But if you want the certification that you've watched this 2 hour lecture, 2 hour and a half lecture, and we can send the certificate to you, but please fill up the form, put up the keyword that we're going to show up on the screen. Pode mostrar aí, Vitória, a palavra-chave a hora que você quiser, tá? Eu não sei se ela tá aparecendo porque eu tô. Ah, tá aparecendo, tô vendo. Then you put up this certificate, you put up this keyword into the forms that we are going to be aware that you've watched the whole lecture. And do the payment, and then we will email you the certificate too, okay? And well, I'll be here if you need any more assistance, just reach out. And if you guys have any questions, send us here in the comments of this video that I can try to answer it right now. If there is no answers or questions being needed, I I let you guys know that my email, my Instagram page will always be available for you guys to send me questions to, okay? Hugs to Nigeria. They're watching us. Thank you guys for the invitation. Thank you guys also for like looking for this kind of knowledge. And I hope it helped, or at least it started to help. And I hope you guys understood my English too. All right, that's it. So, no questions. I'll look at my WhatsApp to see if there's any questions here. All right. So either the lecture was very good and you guys didn't have any questions about it because it's all very explained, or you turned off your English and you're not even listening, just being here present. But anyways, I hope you guys learned a lot with this, and yeah, can't wait for our next trainings. Okay, hugs to you all, and thank you for being part of this activity within the workplace. And keep posted for new opportunities. Bye, guys. See you.